Hello, welcome to Rocky Biochem series. In this video, I will take you through primers for first and then second strand synthesis. Please, if you have not subscribed to my channel yet, kindly support me by doing so. I appreciate all your help towards the growth of my channel. Now, to make us appreciate what we are going to do today, let's do a very quick recap about cDNA library construction. We define cDNA as a complementary DNA synthesized from the reverse transcription of mRNA molecules. So, cDNA library construction involves, first of all, the isolation of mRNA from the cell, and after isolating the mRNA from the cell, then the mRNA will be used as a template for the synthesis of the first cDNA strand. So after the first strand synthesis, we will obtain a hybrid of mRNA and then cDNA. And after obtaining this hybrid, then the mRNA portion will be degraded and then another cDNA will be synthesized to form a double-stranded cDNA molecule. So after being able to synthesize your double-stranded cDNA molecule, then it will be cloned into a vector like plasmid. And after cloning it into the vector like plasmid, then the clones will be transformed into a host like E. coli for it to replicate the clones into multiple copies. Please, if you still have a problem in understanding how cDNA library construction is done, kindly check out my previous video on cDNA library construction. I have put the link to that video in the description of this particular video, so kindly check it out. cDNA synthesis occurs in two steps. First of all, we have to synthesize the first strand and then we have to synthesize the second strand. So the synthesis of the first strand requires certain set of primers. There are some primers that we can use for the first strand synthesis, and there are also some primers we can use for the second strand synthesis. So for the purpose of this video, I am going to take you through the primers we can use for the first strand synthesis and then the primers we can use for the second strand synthesis. For the first strand synthesis, we can use oligo-DT primers, the random primers, the oligo-DT primer linker or oligo-DT primer adapter, and then asymmetrically tailed plasmids. We can make use of any of these primers for the synthesis of the first cDNA strand. I am going to pick each of them one after the other and then explain it in details. Let me start with the oligo-DT primers. So oligo-DT primers are made up of a stretch of 16 to 20 T nucleotides, that are synthesized chemically in the lab. So they are designed to target the poly A tail of the mRNA molecule and serve as a priming site. In the cell, most of the mRNAs isolated are having a poly A tail. So that is why these primers are also poly T. So they will anneal to the poly A tail of the mRNA and then serve as a priming site. So upon annealing to the poly A tail of the mRNA, then the enzyme called reverse transcriptase will then bind to the priming end and then use the mRNA as a template for the synthesis of the cDNA strand. So at the end of the first strand synthesis, we will obtain a cDNA mRNA hybrid. There are some limitations about the use of oligo-DT primers, and these limitations are, first of all, they are limited to only poly A mRNAs. When the mRNA is not having a poly A tail, it will not be suitable for you to use these kind of primers. So for instance, those mRNAs isolated from histones or those mRNAs isolated from other organisms like bacteria lack poly A tail, and hence you can't use oligo-DT primers to prime such mRNAs for the synthesis of the cDNA strand. So aside the use of oligo-DT primers, we can also use random primers. we can also use random primers. Random primers are made up of collection of random sequences, usually says nucleotide sequences long, and they are chemically synthesized in the lab. So they are hexamers, that is, they are 6 nucleotides long, and they are synthesized in the lab. And their synthesis involves the addition of equal amount of A, T, G, and then the C nucleotides to form all possible combinations of hexamers. So at the end, we will obtain about 4 raised to the power 6 possible combinations. That is around 4,096 different hexamers. That is what random primers is all about. Random primers are just a collection of random sequences or a collection of 6 nucleotide long sequences chemically synthesized in the lab. So they are designed to target their complementary regions in the mRNA molecule. So assuming that this is the mRNA we have isolated from the cell and we want to construct cDNA from it, then these random primers will scan through the mRNA and then anneal to their complementary regions. And upon annealing to their complementary regions, then the mixture will be treated with an enzyme called clenal enzyme. Clenal enzyme has only polymerase activity with no nuclease activity. So clenal enzyme will then start this or fill the gaps between the primers and hence lead to the synthesis of our cDNA strand. So random primers are very important when, for instance, the mRNA is not having a poly A tail. So in such situations, random primers can just start priming the mRNA from the middle for the synthesis of our cDNA. And they are also not restricted to only non-poly A mRNA. They can also be used equally for priming poly A mRNA. So the advantage is that they can be used for priming both poly A mRNA and then non-poly A mRNA. But one thing we have to take note about the use of random primers is that after the first strand synthesis, these primers have to be removed from the reaction mixture before we start the second strand synthesis. Now let's move on to the oligo-DT linker or oligo-DT adapter primers. So when you synthesize your cDNA strand, the cDNA strand will be having a blunt end. When I say the cDNA strand is having a blunt end, it means that the two strands are of equal length. And since they are of equal length, it becomes so difficult for you to clone it into the vector DNA. But if it is to be having a sticky end, that is, if the cDNA is to be having a sticky end, it will be so easy for it to be cloned into the vector into the vector DNA. So the genetic engineers sat down and then they sought it relevant to find ways and means of getting or making the cDNA possess a sticky end in order to make cloning so easy. So that is why they proposed the use of oligo-DT linkers or oligo-DT adapters. So let me start with the oligo-DT linkers. Oligo-DT linkers are just oligo-DT linkers is just a combination of oligo-DT sequences and then a linker sequence. A linker sequence is just a short double-stranded DNA sequence that possesses a restriction site within it. So the registration enzymes can be used to cut the linker at the registration site to generate a sticky end. And as for the oligo-DT adapters already have sticky ends, and hence there is no need for the use of registration enzymes to cut to generate the sticky ends. So as I said, the oligo-DT primers possess registration sites within them, and hence by using registration enzymes, they can be cut at those regions to generate sticky ends. So the mechanism of action of oligo-DT primers is similar to that of oligo-DT primers. The mechanism of action of oligo-DT primer linker is similar to that of normal oligo-DT primers. So first of all, the oligo-DT primer linker will anneal to the poly A tail of the mRNA. The oligo DT region will anneal to the poly A tail of the mRNA, and then the reverse transcriptase enzyme will bind and use the mRNA as a template for the synthesis of the cDNA strand. So at the end, we will obtain our cDNA mRNA hybrid. So by obtaining this, then it can be treated. The mRNA cDNA hybrid can be treated with a restriction enzyme to cut the sequence at the linker region in order to generate a sticky end. But one limitation about the use of the oligo DT primer linkers is that in situations whereby a restriction site is also located within the normal mRNA sequence, the restriction enzyme can also cleave at those regions, and due to that, some DNA sequences or some mRNA sequences will be lost. That is, the restriction enzyme can cut at both the linker region and also cut within the normal sequence, and by so doing, some key sequences or some important sequences can be lost. And one thing we should also take note is that oligo DT primer linkers is suitable for only poly A mRNA. We can't use it for non-poly A mRNAs. So in order to overcome the problem whereby the restriction enzyme can cut within normal sequences and then bring about sequence loss, the genetic engineers also propose the use of adapters. As for adapters, they already have sticky ends, and hence there is no need for any restriction enzyme to cut and then generate the sticky ends. So upon priming, then the reverse transcriptase enzyme can use the mRNA to synthesize as a template to synthesize the cDNA strand, and at the end the molecule will already possess sticky end, and it will make cloning so easy. And also the problem of sequence loss has been overcome by using the adapters. Now, aside the use of the oligo DT primer linkers and then the oligo DT adapters, we can also make use of a certain modified version of the oligo DT adapters or the oligo DT primers called asymmetrically tailed plasmid. So asymmetrically tailed plasmids are modified version of oligo DT primers, but this time around the oligo DT primer has already been ligated with the vector. The vector has already been ligated with the oligo DT primers. So by the end of the cDNA synthesis, it would already be inserted into the vector, and there will be no problem with cloning since the plasmid has already been ligated with the cDNA. So let's understand it this way. The primer, the oligo DT primer, is already attached to the plasmid, in which we will clone the cDNA into it. So at the end of the whole cDNA synthesis, we will end up having our cDNA that is already cloned into the vector, and this makes cloning very, very efficient, and there is no going to be any problem with cloning our cDNA into the plasmid DNA. Their mechanism of action is similar to that of the normal oligo DT. So the oligo DT sequences will anneal to the poly A tail of the mRNA, and the reverse transcriptase will use the mRNA as a template for the synthesis of the cDNA strand. So these are basically the different kinds of primers we can use for first strand synthesis. Let me continue with primers for second strand synthesis. As I said, the synthesis of the cDNA used for cloning occurs in two steps. We have to first of all synthesize the first strand, and then we have to degrade the mRNA of the hybrid and then replace it with another cDNA strand to get a double-stranded cDNA molecule that we can use for the cloning. So for the second strand synthesis, we can make use of different types of primers, and they include self-priming. We can make use of self-priming. We can also make use of priming with RNase H enzyme, or priming with oligonucleotides, or priming with oligo DT primer linker or adapter. And we can also use random priming. I am going to take each of them one after the other and then explain them in details. So self-priming harnesses the ability of reverse transcriptase to create a hairpin loop at the end of the first strand synthesis. So when you use the enzyme called reverse transcriptase to synthesize the cDNA strand, as I discussed with you in the first strand synthesis, at the end of the first strand synthesis, there will be hairpin loop formation. The hairpin loop formation occurs as a result of some intramolecular bonding. So, because of the hairpin loop presence, the mRNA cDNA hybrid that has the hairpin loop can be treated with alkali for the alkali to degrade the mRNA. When you treat mRNA with alkali, that is a base, they are not stable and hence they will be degraded, leaving only the cDNA strand with the hairpin loop region. So by having this hairpin loop region, then DNA pool can bind to the 3' OH region of the hairpin loop and then use it for the synthesis of the cDNA strand, and at the end we will obtain a cDNA double-stranded cDNA molecule which still has a hairpin loop. So we have to find ways of degrading the hairpin loop, and hence we will treat it with the enzyme called S1 nuclease. S1 nuclease enzyme has the ability to cleave single-stranded DNA molecules. So when it binds to the hairpin loop region, it will cleave it off, leaving us with a double-stranded cDNA molecule. So this is basically how self-priming occurs. Our second strand synthesis, we have what you call RNase treatment or replacement synthesis. So what we are going to do here is that we are going to treat the mRNA cDNA hybrid obtained from first strand synthesis with the enzyme called RNase. The RNase has the ability to degrade mRNA partially. That is, it will degrade the mRNA partially in such a way that there will be only gaps created in the mRNA. It will not completely degrade the mRNA, but it will rather create some gap within the mRNA. So by having this mRNA with gaps within it, then we can make use of the enzyme called clinal enzyme. Clinal enzyme only has polymerase activity, and hence by using it, it will fill up the gaps within the mRNA and then bring about the synthesis of another cDNA molecule. So at the end, we will obtain a double-stranded cDNA molecule with the help of the clinal enzyme. Now let's move on to oligonucleotide priming. Oligonucleotide priming harnesses the ability of an enzyme called terminal deoxynucleotide transferase, that is TDT for short. The ability of this particular enzyme to add a stretch of the same nucleotide sequence to the 3' end of the cDNA molecule. So TDT can add a homopolymer, that is, it can add similar or the same nucleotide sequence to the end of the DNA molecule, to the end of the cDNA molecule, and by adding it to the end of the cDNA molecule, then we can also make use of this particular primer called, we can make use of oligo primer for it to anneal to the stretch of C nucleotides added by the TDT enzyme, and by doing so, then DNA pool 1 can be used to catalyze the synthesis of the other cDNA strand. So to conclude the whole of this, we have to first of all get our mRNA cDNA hybrid as we obtained from the first strand synthesis, and after that we treat it with alkali to degrade the mRNA strand, and then the enzyme called TDT will add a stretch of C nucleotides to the 3' end of the first cDNA strand, and after that oligo primers can be used to anneal to the C nucleotides added by the TDT, and then DNA pool can be used to catalyze the synthesis of the other cDNA strand. So this is how oligonucleotide priming occurs. Now let's move on to oligo primer linker or oligo primer adapter. So here an oligo adapter or linker is used in the that we use in the first strand synthesis is going to be our priming site for the second strand synthesis. So as I said, the oligonucleotide, as we know from the first strand synthesis, is having the oligonucleotide is having a sequence at the end that we call linker sequence, and the linker sequence possesses the restriction site that will be cut to generate the restriction site. So it functions by, first of all, annealing to the poly A tail of the mRNA molecule, and by annealing to the poly A tail of the mRNA molecule, then the reverse transcriptase enzyme will bind and then catalyze the synthesis of the cDNA molecule, and at the end of the first strand synthesis we obtain cDNA mRNA hybrid. So but after getting your cDNA mRNA hybrid, then we will treat the hybrid with alkali for the alkali to degrade the mRNA, and after degrading the mRNA with alkali, then the 3' end of the linker region will serve as a priming site for DNA pool 1 to bind and then catalyze the synthesis of the other cDNA strand. So at the end we will obtain a double-stranded cDNA molecule. So we have to take note that the priming is done by the 3' OH end of the linker sequence. The same thing will happen for adapters, because adapters and linkers are similar, just that linkers do not have sticky ends, whereas adapters have sticky ends. Now let me move on to random priming. This is similar to what happens in the first strand synthesis. So the random primers, that is usually 6 nucleotides long, is added to the reaction mixture after treating the mRNA cDNA hybrid with alkali to degrade the mRNA portion. So after degrading the mRNA portion by alkali treatment, then the random primers will anneal themselves to their complementary regions within the cDNA strand, and then use it as a template for the synthesis of the other cDNA strand. So clinal enzyme binds, and then the clinal enzyme will use the cDNA as a template for the synthesis of the other cDNA strand. So at the end we will obtain our double-stranded cDNA molecule. So this is basically what you have to know about primers for first strand synthesis and then the primers for second strand synthesis. Thank you for watching this video. Please, I hope the video has been so helpful for you. Kindly subscribe for me and also share with your mates for me as well. Thank you very much. See you another time.